Hey guys, so here we are in our next section on DNA and RNA and proteins. So remember, when we talked last, we talked about how molecular biologists believe that information is transferred in that order: DNA to RNA to proteins. Now we learned that DNA copies itself through a process called replication, that DNA can be then transcribed into RNA, so transcription, change it into RNA. And now we're going to talk about the next process where that RNA is actually translated to form proteins. Our key concepts: translation converts an mRNA message into a protein, aka polypeptide. Polypeptide is a fancy word for protein. And mutations are changes in DNA that may or may not affect the phenotype. And phenotype, again, are physical characteristics. We learned that in the last presentation. So your physical characteristics can be affected by mutations in our DNA. Our vocabulary, we've got translation, codon, anticodon, mutations, mutagen, gene mutation, and chromosomal mutation. Our essential questions: what is translation? How does translation convert an mRNA message into a protein? What are mutations? What are the effects of both gene mutations and chromosomal mutations? Now, we need to talk about mRNAs. Now, mRNA contains the message for actually building the protein. And so a specific sequence of bases in that RNA carries the direct form of a chain of amino acids. And those sequences of bases in the mRNA is genetic code that needs to be translated. So the process of decoding an mRNA message into a protein is translation. Now the four bases in our RNA, and remember, our four bases in RNA are different because they're not the same. We have a U instead of a T in RNA. So the four bases are A, C, G, and U, and act as like letters of our code. Now we read each code 3 letters at a time, so those form our words. So each word is 3 letters long. And then each word corresponds to a single amino acid. And remember, amino acids are the pieces that we need to bring to build our protein. So translating the code. So each 3-letter word in mRNA is known as a codon. And some codons serve as start and stop signals for protein synthesis or making a protein. So some of them say start, so start building protein. When it's long enough, it hits a point where it says stop, so we know the protein is complete. And we can use the codon chart to determine the code, which we'll show you in a little bit later. So if we look at this, here's my segment of mRNA. And again, we read each word 3 letters at a time. Each 3-letter word is a codon. So the first one we have AUG. AUG stands for methionine, which is our amino acid that's going to build part of our protein. And that's also the code for start. Then we have the next 3 letters, CUU, and that corresponds with an amino acid called leucine. And that's going to be the next piece of our protein. So how do we actually read the codon chart? So here's a codon chart. The genetic code matches each codon to its amino acid or function. And basically, we just look at the 3-letter words. So the first thing you're going to see is that over on the very, very side, it says first base. So if we wanted to find our first base, which was our first codon, first word, AUG, we're going to look for A down here on the left side. You're going to find where the green A is, okay? So there's our A. Now we're going to look up top where it says second base. So you're going to find it up top. Okay, so we have our first one over here, second one here. Okay, so you're going to find your U, and you're just going to bring your finger down to match it, and this one over. So you'll be in that box, and you can look in there. Then we want to find our third base, which is over on the other side. Okay, where you see where it says third base. So we said we're going to find the codon AUG. And if we look and we find that right in that box, AUG, we'll see methionine. That is our first amino acid. There's another type of codon chart that looks like this. Maybe you'll find this a little bit easier to look at. So we start in the middle of the circle, and we find our first letter. There it is, A, right in the middle. Then we look in that same piece of pie, okay, that first quarter of the pie, and we're going to find the U. which is just a little smaller sliver. So now you're going to be in that ring. So right underneath that on the third ring, you're going to look for the G. You'll find the G, and again, that corresponds with that amino acid methionine. So how is translation done? Well, mRNA is transcribed in the nucleus and then enters the cytoplasm. So remember, we go from DNA to RNA, and that's transcription. Then we got our mRNA message, which moves out into the cytoplasm and finds the ribosome. On the ribosome, translation begins at the start codon. Start is always AUG. Okay, so inside we've got our little machine, which is our basically like our ribosome is our factory for building our protein. And you can see there's 3 little slots there. Those slots correspond with our 3-letter word or 3 nucleotide codon. Okay. So each codon then attracts a thing called an anticodon, the opposite code. So if I have AUG, my anticodon, my A is going to go with a U. Okay, my AU, my U is going to go with the A, and then my G is going to go with the C. Those are my anticodon, the opposite, the complementary codes. So each codon attracts an anticodon, the complementary sequence of bases on the tRNA. So the tRNA carries that anticodon. So this is how it actually brings the correct amino acid. So each anticodon then holds the amino acid on this tRNA, the transfer molecule, and brings it on over. So each tRNA carries one kind of amino acid. And the match between codon and anticodon ensures that the correct amino acid is added to the growing chain. There's a little picture there for you. So how translation is actually done, the process keeps going until the stop codon is reached. When it says stop, the newly formed chain and the mRNA molecule are released from the ribosome. That newly formed chain of amino acids is a protein. Remember, proteins were made out of small little units called amino acids. So we have different types of mutations. So again, mutations are heritable changes in genetic information, and most mutations happen during DNA replication. Environmental conditions may increase the rate that mutations happen. Now we have these things called mutagens, and mutagens are chemicals or physical agents such as UV rays and chemicals in the environment that can actually cause mutations to occur. We have 2 categories of mutations we're going to talk about, gene mutations and then chromosome mutations. So first thing we're going to talk about are gene mutations. Gene mutations involve changes in one or a few nucleotides. And there are 3 types of gene mutations we're going to talk about: substitution, insertions, and deletions. So substitution. Substitution happens when one nucleotide is changed into a different nucleotide. It may affect only a single amino acid and may have no effect at all on the organism. So if we look down here below, we have our normal DNA. So it's going from DNA to our mRNA message to our protein. And our protein is made out of the amino acids leucine, GLU, SER, and ASP. Okay. Then we have our next one. This is where our mutation, we can see over here, our letter in our original DNA has been changed. Instead of it being a C there, it's now an A, which ultimately changes our mRNA message. Okay, so instead of coding for GLU when it's making that protein, it's now coding for the amino acid ASP. So our next types of mutations are insertion and deletion. They're also called frame shift mutations. These mutations insert or delete a nucleotide in a DNA sequence. And these can change every amino acid that follows the point of mutation. And they can alter or change a protein so much that it's unable to perform its normal functions. So we looked at this, it's a frame shift mutation. So what happens is something gets inserted or deleted. So when you're reading it 3 letters at a time, you had one get deleted here, those 3 letters now change. They either get pushed down or brought forward. So you're reading them differently. So it codes for different amino acids. So change in the order of which those codons are read changes that resulting protein. And again, so here we are. We had started here. We're reading it frame by frame. But now all of a sudden, it's changed. If we change the reading frame, now we're starting with this letter instead. Let's say this one gets deleted. We're starting here, and then we're reading them 3 letters at a time. So it's changed the way the whole thing gets read. Now we've got chromosomal mutations. Now chromosomal mutations produce changes in the number or the structures of chromosomes. And if you remember, chromosomes are tightly compacted, big chunks of DNA. So if you're affecting a chromosome, you're affecting a whole heck of a lot of DNA, not just a small little piece like on a gene. So there's 4 types of chromosomal mutations. We've got deletion, duplication, inversion, and translocation. In deletion, it sounds just like the word. It involves the loss of all or part of a chromosome. Duplication sounds just like what it is. It produces an extra copy of all or part of a chromosome. Inversion actually reverses and changes around the parts of a chromosome. And then translocation occurs when part of one chromosome breaks off and attaches to another one. So if you look at this here, I would like you to copy this in your notebooks. These are what these chromosomal mutations look like, and you can look at the letters. So we start with our normal chromosome there. The first one, you see in deletion, we've removed the letter B completely. In the second one, we've added a letter B, so it's duplication. In the third one, the chromosome kind of gets all messed up and coiled up on itself, and it changes the order in those genes. And then lastly, 2 chromosomes get stuck together. And when they get stuck together, when they pull apart, some of the chromosomes get flip-flopped. And again, please copy this because you may see a picture of a chromosome and they'll ask you which kind of mutation it is. The effects of mutations vary. So chromosomal mutations, because it's a lot of information, there's a lot of DNA on a chromosome, they tend to have a big effect on the organism. An exception to this is genetically modified things, like we, as people, genetically modify a lot of foods that we make. Like in plants, we add extra chromosomes to plants to make them stronger, ripen faster, have more nutrients, all sorts of things. Keep them on the shelf life longer, all sorts of different things. So genetically modified foods have big changes in their chromosomes. Gene mutations may have various effects. Some may be silent, which means it doesn't do anything. Some of them can alter or change proteins so much that it can be useful. So this is called like adaptations, when things have changes that actually help them. But also we have some negative sides. They can disrupt the gene's function or dramatically change the way the protein works, causing genetic disorders. And there's different things out there that cause genetic disorders, one of which is called sickle cell. Sickle cell causes the protein that holds our red blood cells in a round shape that's not present, so it makes the red blood cell have this little sickle, like a little half moon shape. And so it's really stretchy and breaky and sticky instead of being a nice smooth red blood cell. And it's a very common thing to have sickle cell. And actually sickle cell in some environments is very beneficial, because if you have sickle cells and if you have a few sickle cells, not all sickle cells, you are immune from getting malaria, because the malaria transmitted by mosquitoes does not attach to a sickle type of a red blood cell.